join code with the chat. Our class join code is FNNJ5K. FNNJ5K. I'm also going to share the link with the chat as well. You're going to click on that link and you're going to create a profile. Please don't forget your username and password for this profile, because we will be using this activity, this resource throughout the rest of the course. And what I want to do now is stop sharing my screen, and I want one of you to volunteer to share your screen so that we can walk through this activity and you all can see what it looks like from the student side. So would someone please volunteer to share their screen for us? Thank you, DeMarco. Yep, it's that first one there, the Explore Learning. If you probably will need to create your own account. So click enter our, where it says class code. Our class code is FNNJ.
Oh, bro, chill.
5K. Good. So you probably need to create an account unless you've already done it for another class.
I think I already got an account.
Okay. Go, DeMarco, go back. I don't think you're on the right screen. No. Go back to the. Why did you stop sharing? Share your screen again for us. So just hit the back button there, yeah. So where it says I need to create an account, and I think it's green on your screen. Yep, click on that. Yeah. Enter your personal info. You don't need to enter an email address. That's not necessary. It could be helpful. But again, please do not forget your username and password. So if you need to write it down somewhere or type it into a note on your phone, that may be a good idea for some of you who may be more prone to forgetting things. That's also a good idea. Well, he just made his 1, 2, 3, 4, 5, 6, 7, 8, 9, but you can make it your student ID number. We don't have a registration for this username and password. Please make sure you enter. Okay, I'm not sure what happened. Can you just try to log in, DeMarco? Okay, so something went wrong when you tried to create your account. I'm not sure what you did, but try it again. I'm glad we're having this issue because that means that other people may have it as well. Ashley says she was able to get in. So Aaron too. So enter the code, it's 5NN. Yeah. So I don't try to create an account again because something went wrong the first time. What is going on? DeMarco, this is not something is not going right with yours. Okay, let's have you stop sharing and let's have somebody else, Aaron or Ashley or Kyrie, somebody else share for now. And DeMarco, you and I can work on it later. Someone who has been able to get in, please share their screen with us, please.
Come on now.
Okay, thank you, Kyrie. All right, great. So when you get in right now, there's only one option for the activity. Today's activity is called Building DNA. We won't have time because today is a shortened lesson to actually do it during class. But the directions are can be found in two places. So number one, you see on the left side, it says drag and join nucleosides and phosphates to build the left side of the DNA molecule. So Kyrie, what you should do is drag one of those. You see where it says nucleosides, that red A? Yeah. Yep. Drag one of those. And just into the middle. Drag it into the middle. Yeah. All right. Now grab a phosphate, which is that blue circle, and put it on the, yeah, good. Now grab another nucleoside and connect it to that same phosphate. From the top. So like put it in the top right. Yep. Okay. Good. And you want to just keep going in that way. Keep adding them. Good. Okay. Now pause there, Kyrie. You see where it says lesson info at the top? Scroll up. At the top of your right under your name, it says lesson info. Yeah, click on lesson info. Now you have two options there. One of them says vocabulary sheet, the other says student exploration sheet. We won't be working too much with the vocabulary sheets. We will be working primarily with the student exploration sheets, and you may hear me refer to these as the Gizmo worksheets. So what I want you to do now, Kyrie, is click on Google Doc. Do you see Google Doc right there? Yep, click on that. And then click on Make a Copy. So this worksheet is what you actually need to complete in order to receive credit for the Gizmo. It's got questions here, and you just type directly into this to answer the questions. And as you go, it'll give you instructions about what you should be doing in the Gizmo. So just like Kyrie just dragged those things from the right into the middle of the screen, it'll provide the directions to tell you what to do next. So as you do the worksheet, it gives you the directions about what to do. Now, what I want you to do, Kyrie, is change the name of this document. So click in the top left where it says copy of, blah, blah, blah. Delete the copy of and type your name. Okay, well, that's fine. So Kyrie Little, building DNA. Excellent. And then do you see that little folder there, Kyrie? That's just to the right of your, no, not that. Go back. Do you see where it says, do you see the star at the top that's right next to the Kyrie Little building DNA? There should be a folder that comes up next to that in a couple, maybe a few seconds as it finishes loading. Or click on the cloud, yeah. Okay, that's not it. Okay, so instead click on File. Oh, there's the folder right there. Yeah, click on that folder. That folder is, it means move. So now we're gonna move this document into his biology Google folder. So Kyrie, scroll down and find your biology Google folder.
I ain't got no biology.
Ah, okay. So that's what you need to do. You need to make your biology Google folder. But once you all have gotten to this point where Kyrie is, that's what you'll do. You'll save your worksheet into your biology Google folder. And that is how, essentially how you'll submit it to me. All right, so thank you, Kyrie, for sharing your screen. We are good to move on at this point. If you have any questions about this process, please do not hesitate to reach out. I'll probably put a brief video up explaining exactly how to access those tools, and you all will be able to watch that video and you'll be able to walk through it yourself. But Kyrie, what Kyrie just did was 100% correct. So from there, I will share my screen and we'll jump into the notes for the day. We've got about 29 minutes left, which should be just the right amount of time. Okay. So at this point, you should have your notes out, please. And you should write down the title of today's lesson, which is DNA Replication. You should write down today's date, which is October 7th, 2020. And you should write down that we are covering Unit 5, Day 2. You don't need to write down the objective or the essential question, but I'm going to go ahead and read them anyway. Biologists will be able to explain the process of DNA replication, which copies DNA during synthesis of interphase. So if we remember the cell cycle that we talked about in Unit 3, the cell cycle has several parts. One of the longest part of the cell cycle is called interphase. And interphase is also made up of several parts. We've got G1, S, and G2. G1 being gap 1, then S is synthesis. That's where the DNA replication takes place. And our essential question for the day that we will seek to answer is DNA replication an essential process for living organisms? Why is DNA replication an essential process for living organisms? So the question, why does our DNA need to be replicated? I want someone to try to give me some help here. Tell me what they think. Why does the DNA need to be replicated? Why do we replicate DNA? You can type it into the chat or you can unmute yourself and give me an answer. Again, think back to what we talked about during our unit on the cell cycle. Let's see. Jaden says 2 new daughter cells must contain the same genetic information. Good, Jaden. That's basically correct, right? So we need to replicate the DNA because when we reproduce, when we make new cells, those new cells need DNA as well. So you should write this down. Again, when you see something that's highlighted, that's what you should be writing down in your notes. So the question, why does our DNA need to be replicated? The answer, before a cell can divide during M phase, there must be a copy of the DNA for the new cells. Each new cell needs its own copy of the DNA. So that means we need to copy the parent's DNA. All right, so before the cells can divide during M phase, which is either mitosis or meiosis, there must be a copy of the DNA for the new cells. Now, DNA replication is a lot. There's a lot going on in this process. I'm not going to get into it all. If you're interested in this process, I would encourage taking the AP biology class where we do break this process down. But the most important takeaway, we start off with old DNA, and we eventually end up with 2 new strands of DNA. So we start off with old, we end up with 2 new strands of DNA. Now, one thing to take note of, we've got ACE here, ACE there, ACE there, ACE, ACE, ACE. What do all of these ACE endings mean? When I see the ending ASE, what should I be thinking about? When I see the ending ASE, what should I be thinking about?
Enzymes.
Enzymes. Excellent, Jaden. Yeah. So enzymes represent, I'm sorry, ASE represents enzymes. So all of these ASE endings that we're seeing, DNA primase, DNA ligase, DNA polymerase, helicase, topoisomerase, they all end in ASE because they are all enzymes. And what do we know about enzymes? What do enzymes do for us? What is the role of an enzyme? What is the role of an enzyme?
Chemical reaction.
What do they do for chemical reactions, Jaden?
Hold on, I'm checking my notes.
Good, excellent. Jaden is modeling what you all should be doing. Hopefully you have access to your notes so you can look back. Enzymes have a very important function. I'm going to go ahead and answer that question now, Jaden, so we can move on. But
Okay.
I'm proud of you for having your notes out. Enzymes speed up chemical reactions. Okay, so reactions would almost always happen, without any help from enzymes, but enzymes speed them up so that our bodies can take care of these processes much more efficiently. Okay, so we need these enzymes so that this process can happen quickly. Another question, where is this process of DNA replication occurring? Where does the DNA replication occur? The big hint is if DNA is involved, then where is this probably happening? Okay, excellent job, Jaden. So we know that it happens in the cytoplasm for one specific type of cell. But in another type of cell, where would it be happening? So that was 50% of the answer. What's the other 50%? Okay, so in prokaryotic cells, yep, that's where we have it in the cytoplasm. What about in eukaryotic cells? Where does DNA replication happen? In the nucleus. Excellent job, Jessica. So they tag team. Thank you, Kyrie. Good. So wherever we find DNA in the cell is where DNA replication occurs. So like Jaden told us, in the prokaryotic cell, this takes place in the cytoplasm. And then like Kyrie and Jessica told us, in a eukaryotic cell, this takes place in the nucleus. So wherever you can find DNA in the cell is where DNA replication will be taking place. Okay. So a little DNA joke for you all here. We've got a prokaryotic cell saying, you tried to copy my DNA, but you did it backwards. And then the eukaryotic cell responds by saying, and? See what I did there? DNA backwards is and. Yes, no? Okay. All right, no laughs. Tough crowd today. All right. Okay, so that was a little joke interlude for you all, but moving back to the notes. DNA replication is a semi-conservative process. Semi-conservative. What we mean by this is that half of the original parent cell's DNA is saved, meaning half of it is conserved. And the other half is used in the new, or the other half comes from a new source of DNA. So in this image here, we've got an original template. We've got the parent DNA, which is all blue. But as we copy it, half of that blue remains, and the other half is red. It's coming from another source. It's newly synthesized. It's newly made. And this means that each of the two new strands of DNA has half of the old strand and half of the new strand. So please make sure you're writing down that definition of semi-conservative. Half of the original parent cell's DNA is saved, meaning it is conserved, and it's used in the new daughter cell's DNA. Got 17 minutes left. All right. So the process of DNA replication can sometimes seem a bit confusing, but we're going to try to break it down by using some common terms that you all are familiar with. The first step is an enzyme called helicase coming in and unzipping the double helix. So we remember DNA is basically like a ladder that has been twisted into a spiral. So this helicase is going to come in and it's going to clip the rungs of that ladder. It's going to break the hydrogen bonds that keep the rungs together, going to separate those. And so in effect, it's unzipping the DNA. So something that was connected at first is now getting unzipped and separated. Starts off like this. Now it's coming and being separated. And we can see in that animation pretty much what's happening. Okay, the second step, another enzyme, this one is called DNA polymerase, comes in and adds complementary base pairs to each of the exposed strands. And when we say complementary base pairs, what we're talking about is that adenine, which is your A, is complementary to thymine. So anywhere I see a thymine, I need to add an A. Anywhere I see an adenine, I need to add a T. They're complementary. They always go together. They're matched up. Same thing with guanine and cytosine. Whenever I see a G, I need to add a C. Whenever I see a C, I need to add a G. That's what we mean by complementary. And this enzyme, DNA polymerase, is in charge of making sure that they get added in the proper places. We can see that taking place here. Where there's a G, we add a C. Where there's an A, we add a T. Here on this C, we need to come in and add a G. Here on this T, we need to come in and add an A. And the same thing is happening on both strands. The next step is that another enzyme, so we can see a lot of enzyme involvement here, another enzyme called ligase reforms the new hydrogen bonds between the complementary base pairs. So we unzipped, we add new base pairs to make two strands, and then ligase comes in and it zips them back up together. It adds hydrogen bonds to zip them together again. Okay, and then the last step is probably the most important. And this is when another enzyme, this one also is called DNA polymerase, comes in and proofreads the strands to make sure that there are no mistakes. And of course, mistakes they show up as mutations. Okay, so here's what the process looks like in simplified format. Again, DNA really looks like a ladder. We've got two backbones. In the middle, there are nitrogen bases that are connected by hydrogen bonds. We start off with these being connected. Then Helicase comes in and separates them. So now we've got one strand over here, one strand over here. Then we're going to come in and add new bases to each of these old strands. And eventually we end up with two new DNA molecules, each of which is made up of half of an old strand and half new strand. So I'll let you all process this image. Okay. So we'll watch this quick video and then we're going to practice doing some complementary base pairing.
DNA is the molecule made up of two strands.
Ah, sorry.
DNA is the molecule made up of two strands. twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes the small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so let's practice very quickly. If I've got a strand of DNA that reads T, A, C, C, G, A, T, C, G, C, A, can you all tell me what the complementary strand will read? Keep in mind, A complements T and vice versa. C complements G and vice versa. So meaning T also complements A, G complements C. What this looks like, if there's a T, then I need to put an A there. If there's an A, then I need to put a T there. What should I put if there's a C? What should I put to complement C?
G.
G, yep. Then what?
G.
Yep.
C.
Then
T. A. G. C. G. T.
excellent. Yeah. So what we just did, and who was that, DeMarco or Kyrie?
Jaden.
Oh, that was Jaden. Good job, Jaden.
Okay. Yeah.
So what we're doing is we're complementing each of these bases. So anytime I see a C, that means the complement has to be a G. Anytime I see a G, that means the complement has to be a C. And anytime I see an A, there needs to be a T. And anytime I see a T, there needs to be an A. Let's try another one. What should I put first?
Go ahead, Kyrie. Got you. A T G C T G C A T C A G.
Excellent.
Yeah, boy. Yeah. That's right, boy.
Good job. All right, so we're going to continue to practice with this as we go, but I want to push you all just a little bit further. This is the same strand that Kyrie just complemented for us, but I want to go from this strand to RNA. We've got to keep in mind that RNA does not have T's. It has U's instead. So anywhere I would put a T for RNA, I need to put a U. So, for example, A is typically complemented by T, but RNA does not have T's. It has U's, so I need to put a U there. What should I put here?
A.
An A.
A T.
Okay, so if this were DNA, then yes, I would put a T here, but this is RNA.
Oh, a U.
So I need to put a U.
A. A C. G. U.
Okay, so remember T complement. What is T complemented by?
A.
A. Yeah.
That's confusing.
We're going to continue practice with this.
C. G.
C, G,
Well, that's U.
U.
That's
Good.
A.
A.
G. G. U. And then C.
Excellent. Right. So we see that RNA does not have any T's. Any place that I would have put a T, I needed to put a U. We're going to practice more with this. The gizmo walks you all through this, but we are now at the end of our time today since this is a shortened class period, and I will let you all get ready for fourth block. Please, again, let me know if you need any help finding those gizmos. And I hope you have a good Wednesday. Talk to you all tomorrow.